their colleagues, their students, their friends, it is on behalf of the Duve Institute and of the health sector of UC Louvain, it is my great pleasure to welcome you today at the 4th Duve Memorial Lecture. This lecture series was initiated in 2013, the year of the death of our founder, Christian de Duve, who, as you all know, won the Nobel Prize in 1974 for his discovery of lysosomes. This lecture series was established to honor de Duve's memory. It is held every other year. It is meant to be prestigious and to give the word to distinguished international speakers in the field of biomedical sciences. You can see on the next slide that we had 3 lectures so far, with Michael Hall in 2013 talking about the discovery of mTOR, with Andrea Balabio in 2015 talking about the biogenesis of lysosomes, and David Sabatini in 2017 talking about mTOR and lysosomes. So you can see these are very big names. And this year, today, we are fortunate enough to add another very big name to this series. I am very pleased to introduce Carl-Henrik Heldin from Uppsala. Kali is professor of molecular cell biology at Uppsala University, where he spent most of his scientific career. Sorry, I should have switched to the next slide. Yes. He was director of the Uppsala branch of the Ludwig Institute for Cancer Research during more than 30 years until his retirement in 2017. He also served the international scientific community by his highly appreciated role in different key positions at renowned organizations. And to name only a few, these are organizations like EMBO, where he is currently chair of the board, and the ERC, European Research Council, which he served as board member between 2005 and 2015, and as vice president in the last 4 years, 2011-2014. Kali is also currently chairman of the board of the Nobel Foundation. The scientific interest of Kali is in the study of growth factors. This should not go like that, yes. Growth factor, initially PDGF, and then he switched to TGF, tumor growth factors, which is a pleiotropic growth factor or cytokine playing a role in multiple and numerous processes from embryogenesis to inflammation to tissue repair, cancer, and autoimmunity. So this led him to touch many different aspects of biology with a particular interest in cancer and also in the signaling aspects of those growth factors. Over the years, Kali has trained a whole generation of scientists working with great success on TGF-Beta and PDGF in different labs all over the world. TGF has become an entire field in its own right, and Kali is recognized as one of the fathers of this field and one of the leading authorities in TGF-Beta and PDGF. His list of achievements is very impressive, with over 500 publications, cited around more than 68,000 times, and an age index, for those of you who like age indexes, of an age index of 138. Very impressive. Despite having reached retirement age in Sweden, Kali is still active in the lab, thanks to the fact that he recently obtained an ERC Advance grant. I'm very pleased that he has accepted our invitation to deliver the 4th lecture, which will focus on TGF, for the Duve Lecture, which will focus on TGF-Beta signaling, a field that Kali has pioneered. So Kali, the floor is yours.
Thank you very much, Benoit, for this kind introduction. It's an honor to give the Christian de Duve Lecture. I'm very happy to be here. I have a very interesting day at the Duve Institute and enjoyed talking to many of the excellent scientists here. I would like to start by showing this slide, which encapsulates what I have been interested in during my career so far. the first 4 years. This is a normal cell who receives signals from outside that controls its proliferation and survival. Which is one of the interests in our lab, stimulates the cell to grow. But there are also inhibitory factors, and TGF-Beta, transforming growth factor beta, which we also have devoted quite a lot of effort to, is one of these negative signaling molecules. It is common to see in cancers that signaling molecules along these myogenic pathways are overactive, or that signaling molecules along this suppressive pathway is lost. And this is almost obligatory in tumorigenesis and accounts for the loss of growth control that characterizes cancer. So I'd like to spend just a few minutes on TGF, although it deviates slightly from the title of the lecture. So TGF is a family of 5 members, A, B, C, and D homodimers, and the AB heterodimers. They bind to tyrosine kinase receptors, of which there are 2 kinds, the alpha receptor and the beta receptor. And the ligand binding induces dimerization of these receptors, which is a key event in the activation of the receptors. So this is because the receptors come close to each other and can phosphorylate each other in trans. And this phosphorylation occurs on specific tyrosine residues. And we devoted quite a lot of time during the 90s to elucidate exactly which of these tyrosines were and how they then initiate signaling pathways of different kinds, and we are listed here. Major persons in this effort was Lars Rönnstrand and Lena Clausen-Welch and Jean-Baptiste Lemoulin when he worked in our lab as a postdoctoral fellow some 15-20 years ago, also took part in this effort. So this is now rather well characterized. But a recent finding we have made is that the receptor, after it has been activated by its ligand, moves to the nucleus, at least part of it does. So this is the nuclear fraction. As you can see, the receptor accumulates in the nucleus with a maximum of 30 minutes. In the cytoplasm, it is also present, of course. If you quantify, you see that around 20% of the receptor moves to the nucleus. And this shows a phosphotyrosine blot showing that the activated receptor is the one that is moving to the nucleus. So we looked at this also with microscopy, and if you want to look at things that moves to the nucleus or rest in the nucleus, you have to be very careful with controls in order to avoid looking at background stains. So we used one antibody against the N-terminus of the receptor and another against the C-terminus, and then we looked at the merging of these colors. And as you can see in this confocal microscopy, there are some yellow dots in the nucleus. Of course, most of the dots representing the receptors are not in the nucleus, but there are some in the sections that passes through the nucleus, and we don't see them. So how does the whole receptor then go back to the nucleus? And we have tried to understand this. It's not trivial for a membrane receptor to... that. So the question is then how the receptor goes to the nucleus. And as is illustrated here, tyrosine kinase receptors are often internalized via clathrin-coated pits. This is true for the PGF receptor as well, and this is necessary to get the receptor to the nucleus. It also then moves through endocytic vesicles retrogradely through the Golgi system and the ER into the nucleus. And this is can be blocked if you block with an inhibitor of this pathway, Heldin A. You see that the accumulation of the nuclear receptor goes down. And also important beta, which is important for the uptake of molecules through the nuclear pores, is also important. As you can see here, the nuclear accumulation is depleted. So what does it then do in the receptor, in the nucleus? Well, we have seen that the receptor interacts with TMF1, which in turn interacts with this SWI SNF complex, which is of interest in cell growth regulation because components of this complex are deleted or mutated in 20% of cancers, acting as tumor suppressors. And we are currently analyzing the data from a ChIP-seq analysis that we just performed in order to understand more precisely exactly which genes are regulated by the receptor in the nucleus. So deregulation of signaling pathways is involved in the loss of growth control of cells in conjunction with tumorigenesis, as I mentioned. So in the case of PGF and PGF receptors, it has been shown that translocation of the PGF-B gene to a collagen gene occurs in dermatofibrosarcoma protuberans. This leads to the production of a fusion protein, which is processed then to a molecule which is similar to PTGF, which then acts in a truly autocrine fashion, driving this malignancy. The receptor can also be seen as fusion proteins, which has been explored here by Jean-Baptiste Desmoulins and many of his colleagues. And this occurs in some rather uncommon leukemias. And the partners that the receptor is dimerized to has in common that they are oligomerizing or dimerizing, thereby causing constitutive activation of the receptor. There are also examples of point mutations in the receptor, for instance, in gastrointestinal stromal tumors, where the alpha receptor is constitutively active. And in glioblastoma, the alpha receptor is sometimes amplified in up to 10% of the cases. So this means that by use of PDGF receptor kinase inhibitors, could treat patients with these diseases. And those with these rather uncommon tumors are actually helped by this treatment, but unfortunately not patients with glioblastomas. However, PDGF has a more general role in solid tumors because it has a crucial effect on cells in the stroma. So in order to elucidate exactly what effect it has, we took advantage of mice which has the TGF-Beta receptor mutated in such a way that in the ATP binding pocket there is a cavity which has been introduced by a mutation. And this means that it is possible to find inhibitors that are very specific to inhibition of only this mutated kinase and no other enzymes. And by use of this, one can then, in a very precise way, explore exactly what inhibition of this particular kinase of the TGF-Beta receptor does. So we obtained this mice from Carl-Henrik, and then we have used them in order to study the role of PDGF-Beta receptor signaling in the stroma. We selected 4 cell lines that are lacking PDGF receptors or have the PDGF receptor at very low levels. 2 of them contain quite high levels of PDGF, the ligand, and 2 rather low levels. And then we grow these cells in the mice, and we treated them either with this very specific inhibitor or with imatinib, which is a kinase inhibitor that is used clinically, which inhibits both the PDGF-alpha and beta receptor, and a few more tyrosine kinase receptors. So we could see that in the cell lines that made quite a lot of PDGF, there is an effect of treatment with a specific PDGF receptor kinase inhibitor. Strangely not by treatment with imatinib, which also inhibits the PDGF receptor, but it also has other effects. I think that is the reason for this discrepancy. And this is true also for the other cell line. I would like to point out that these cells, tumor cells, do not have any PDGF receptor. So this effect is exclusively through the effect on the stroma cell. So what is then the effect? So this is an analysis by immunohistochemistry of sections from the tumors, and you can see here staining for vessels that this is dramatically lost upon treatment with the specific inhibitors. Also, the treatment with imatinib causes less vessels, and this is a quantification. And as you can see, the specific inhibitor has an even more or equally good effect as imatinib itself. We also wanted to see what functionality of the vessels, and therefore we perfused the tissue and looked at the accumulation of FITS-labeled lectin. As you can see in the controls, there is quite a lot of it. It's much less in the treated mice, both with the NIPP1, the specific inhibitor, and with imatinib. And here is the quantification. And there is also an induction of apoptosis in the endothelial cells. If you look here on the red color, which is cleaved caspase 3, on top of the green color, which is the vessels, you see that there is quite a lot of yellow staining, meaning that there is a colocalization. And more in the specific inhibitor-treated mice than in imatinib-treated mice. We then look specifically on the parasite population because PDGF is known to have an important role for parasites, and we could see then that inhibition of PDGF receptor kinase caused a decrease in NG2 carrying parasites as well as in cells carrying the PDGF receptor, and this was more efficient in the specific inhibitor-treated mice than in the imatinib-treated mice. So we are then, in conclusion, we do see an effect of inhibiting PDGF receptor kinase in the stroma cells, particularly if we use a very specific inhibitor. And we do not see the same efficiency if we use imatinib. We think that this is because imatinib, in addition to cause inhibition of the PDGF receptor kinase, also affects the apoptosis machinery, and which has the opposite effect, and therefore the treatment is less efficient. So this has some clinical implications because imatinib has been used to try to treat a number of different solid tumors, often together with other drugs, and the result has been fairly not so positive, more than for certain very specific tumor types. Maybe if one can devise a more specific PDGF receptor kinase inhibitor, the treatment effects would be more substantial. Now coming to TGF-Beta. So TGF-Beta is a prototype for a family of more than 30 structurally related dimeric factors in humans. And these includes then TGF-Betas, activins, inhibins, bone morphogenetic proteins, and growth and differentiation factors. They all act via formation of heterotetrameric receptor complexes of type 1 and type 2 serine threonine kinase receptors. And they all have important roles during embryonal development, and their cellular effects include very often growth inhibition, but under certain circumstances could also be growth stimulatory. Many of them causes matrix production, and they all affect cell differentiation, and some of them induces apoptosis. So this is a schematic illustration of the signaling effects of TGF-Beta. It's made as a latent precursor and need to be activated before it can bind with receptors. And when it has bound to type 1, type 2 receptor complexes, it induces signaling via SMAD molecules. SMAD2 and 3 are phosphorylated and activated by the type 1 receptor, TGF-Beta receptor kinase. Then they form complexes with SMAD4 that moves into the nucleus, where they then act as transcription factors. But there is also non-SMAD signaling pathways, including MAP kinases, P38, JNK, and ERK, as well as the receptor itself can be cleaved, and the intracellular part moves into the nucleus and acts as a transcription factor. And also the activation of Rho and SRC and PI3 kinase are also seen in TGF-Beta stimulated cells. So we have been interested in the role of TGF-Beta in various diseases. It has been well established that TGF-Beta is involved in fibrotic conditions, glomerulonephritis, lung fibrosis, liver cirrhosis, and so forth. And this is probably mainly because of the ability of TGF-Beta to cause accumulation of matrix proteins. In cancer, TGF-Beta has a complicated role. Initially, it's a tumor suppressor because it inhibits growth of most epithelial cells, at least, and many other cell types as well as induces apoptosis. But at later stages, it becomes a tumor promoter. And this involves effects on tumor cells. For instance, induction of epithelial mesenchymal transition, which is connected with increased invasiveness and metastasis formation, and effects on other cell types. For instance, TGF-Beta suppresses the immune system, and it promotes angiogenesis, which also is tumor-promoting events. Why then explore TGF-Beta inhibitors for the treatment of advanced cancers? Well, TGF-Beta is expressed at high levels in most solid tumors, and studies on animal models support the notion that TGF-Beta drives the development of advanced cancers. And TGF-Beta has tumor-promoting effects both on cancer cells and normal cells. And TGF-Beta promotes metastasis, which is the cause of death for more than 90% of cancer patients. So if we want to now inhibit TGF-Beta in advanced cancers, we need to be selective. And we have tried to then elucidate which of the signaling pathways that TGF-beta induces that are tumor suppressive versus tumor promoting. Via SMAD2, SMAD4 complexes, TGF-beta causes cell differentiation during embryonal development and growth arrest. Via SMAD3, SMAD4 complexes, induction of EMT. Via JNK and P38 MAP kinase pathways, apoptosis. And via ERK MAP kinase pathway or AP1 transcription factors, phosphatidylenosyl 3 kinase, the tyrosine kinase SRC, and then this intracellular part of the receptor, it causes growth stimulation and survival and migration and invasion. So we then would like to inhibit the red signals in these signaling pathways while retaining the green ones, trying to selectively inhibit then the tumor promoting pathways, but not the tumor suppressing ones. Another way of dealing with this is, of course, to be more geographic in this selection, which Sophie Lucas has done here in a very nice way by using the interaction between the latent form of TGF-beta and the GARP, and thereby being able to inhibit activation of TGF-beta locally. Because to inhibit TGF-beta systemically is dangerous because it could promote tumor formation and it also could make too much side effects. So to give you, I should emphasize that this is a very pronounced oversimplification. Life is much more complicated. I'm showing you this just to illustrate the thinking behind our approach. And I'd like to tell you a little bit about our efforts now to identify the mechanism whereby these signaling pathways are induced. So to start with the AP1 family to start with SMAD pathways and its interaction with other transcription factors. SMADs can be involved both in growth inhibition and apoptosis, as I have mentioned, but also in invasion and metastasis. And it does so by collaborating with transcription factors of different kinds. And we then performed a ChIP-seq analysis with antibodies recognizing SMAD2 and 3. In cells that had been stimulated with TGF-beta for 1.5 hours, we obtained about more than 2,200 hits. But if we then stimulated cells with TGF-beta for a longer time period, 16 hours, we got even more. Interestingly, very few of these hits were really common between the early time point and late time point. And if we then looked on the genes that we could identify in the early time point, components that are known to be involved in TGF-beta signaling were picked up, which is not surprising. At the later time point, several genes involved in focal adhesions and MAP kinase signaling pathways were seen. We then wanted to look more in detail on this, and we could see that on the genes that was brought down by the SMAD2, SMAD3 antibodies, we could see that the CAGA binding motif, which is not surprising because SMADs are known to bind to this motif, but we could also see often the AP1 motif in the same region. So we then explored in more detail the interaction between SMADs and AP1. And not only do they bind to the same promoters, SMADs also induces certain members of the JON family, like JON itself and JON-B and FOS-B, as you see here, but not other members of the family. So we could then show that in a ChIP-seq analysis, the induction of the SERPIN gene, which is encoding PI1, is completely dependent on JON-B because knockdown of JON-B makes the TGF-beta, makes the binding to this promoter go down. Same is true for LAMP3, but not for MMP2, which is another target for TGF-beta. We could also see that if we look at the mRNA, there is a similar induction after 1 hour and even more after 16 hours by TGF-beta of the SERPIN gene. And if we then knock down JON-B by siRNA, this goes down. Same is true for LAMP3, but not for MMP2. We could also see that if we knock down John B by siRNA, the invasiveness of cells, this is in a zebrafish model, and in the tail here you see that there's much more cells labeled by red fluorescence that are invading the tissue compared to the controls, to the si John B transfected cells. So this is how SMADs interact then with members of the AP1 family, for instance, John B. First, they are induced, and then they act together with SMADs, and then they activate a number of genes involved in migration of cells. So which genes could that include? Well, if we compare the genes that were induced by TGF-Beta after 16 hours in the control versus those cells that were in which John B has been knocked down, these are the controls, we could identify a member of the Wnt family, 7b, which we decided to explore further. And we could see then that Wnt 7b is induced by TGF-Beta as expected, and we could also see that the induction of Wnt B is dependent on John B, because if you treat cells with siRNA for John B, the expression goes down. The same is true if you knock down SMAD4. If we now again use the zebrafish invasion model, you see that breast cancer cells, control cells, show a limited amount of invasion, but if we now transfect John 7b into these cells, there is more invasiveness of cells. This is true also in an in vitro invasion model. Here we grow cells in spheres. If you stimulate with TGF-Beta, you see that cells grow out of the spheres into the collagen around it. If we transfect cells with Wnt B, you see also an enhanced migration, and the combination is giving even more migration. If one adds a TGF-Beta receptor kinase inhibitor, this is suppressed almost completely, and the Wnt is still Wnt transfected cells still migrate a little bit. If we now add a Wnt inhibitor, we see a little bit of effect. So this is a summary of the interaction here between SMADs and John B, and as you can see here, this collaboration induces migration of genes. One example is John 7b, but there is also others. So Marianne Landström noticed that in the type 1 receptor for TGF-Beta, which is also called ALK5, there is a motif which is similar to the consensus motif for binding of the ubiquitin ligase TRAF6. So we then explore the possibility that TRAF6 is involved in TGF-Beta signaling. And in work that was published some years ago, we could show that TRAF6 actually ubiquitinates TAC1, and which then activates MKK3 and 6, which in turn activates P38, inducing apoptosis of cells. So TRAF6, being a ubiquitin ligase, has an important role in these non-SMAD signaling pathways linked to apoptosis. Same is true for JNK, which is another member of the MAP kinase pathway. And here is the data that illustrates that the TGF-Beta induced apoptosis is completely suppressed in cells in which one knocks down TRAF6. I should emphasize that TRAF6 ubiquitinates cells by polyubiquitin chains with lysine via lysine 63, which is not causing degradation, but rather have other effects. So in addition to this well-characterized SMAD signaling pathway downstream of the receptor, we have now to take into account also TRAF6-mediated signaling events. These are not dependent on the kinase of the receptor, but is dependent on ubiquitination, in contrast to the SMAD pathway, which is dependent on the kinase of the receptor. So we then made the observation that after TGF-Beta stimulation of cells, the receptor accumulates in the nucleus, as is shown here. This is using an antibody against the intracellular part of the receptor. If we use instead an antibody against the extracellular part, there is much less there is no nuclear localization. There is some staining, as you can see, but it is not in the nucleus. So we suspected then that this is because of the cleavage of the receptor, and as is shown on this slide, TGF-Beta stimulation causes the accumulation of the fragment which has the size of the intracellular domain of the type 1 receptor. If we look now on the TRAF6 negative binder, this is a receptor which has a crucial amino acid mutated so that TRAF6 can no longer bind. You see that there is no formation of this intracellular domain. And furthermore, this intracellular domain can be ubiquitinated, as is shown on this slide, presumably by TRAF6. So we have this intracellular fragment of the receptor of around 34 kilodalton, but that would still be hanging in the membrane because the cleavage occurs outside the membrane. So there is a need for a second cleavage, and I'm sure you are all aware of Notch being undergoing a similar dual cleavage. The second one via gamma secretase, which is also called presenilin, which is a transmembrane spanning protease which cleaves certain receptor-like molecules in the transmembrane domain. So Shuam-Guday showed that the cleavage of the receptor is dependent on the presenilin molecule, because if you knock it down, you see that the formation of this intracellular domain is not seen. So what does then the intracellular domain of the receptor do in the nucleus? Well, it interacts with the coactivator P300, as is shown here in this co-immunoprecipitation assay, and it also causes expression of SNAIL1 and MMP2, which are genes involved in invasion, as is shown here, after 12 and 24 hours. And these open bars are cells unable to cause the intracellular domain of the receptor because of the lack of TRAF6 binding. So it appears that the intracellular domain of the receptor in the nucleus, in collaboration with P300 and possibly other molecules, drives an invasiveness program. And this is illustrated by this experiment, in which cells were grown on a collagen gel, and then they migrate in response to TGF-Beta through the gel and to the other side of a filter. If one instead transfect the transmembrane mutated receptor, which cannot be cleaved by gamma secretase, there is no TGF-Beta induced migration invasion of these cells. Remarkably, this nuclear translocation of the intracellular domain we see in response to TGF-Beta stimulation in these PC3U cells, which is prostate cancer cells, and some other epithelial cancers as well. But we don't see it in normal cells. This is prostate epithelial cells, normal, and you don't see that the receptor is accumulating in the nucleus. We don't understand why there is a tumor specificity here. It could be as trivial as there is more protease activity in the tumor cells, but we don't really know. So if we look now on the TGF-Beta signaling mechanisms, we have, of course, the SMAD pathway, which is very important. We have the TRAF6-mediated activation of P38 and JNK MAP kinase pathways that links to apoptosis, but also to invasion. And we have the formation of the intracellular domain of the receptor, which certainly induces invasion. If we look now more in detail on the non-SMAD signaling pathways, in addition to those I already mentioned, TGF-Beta activates PI3 kinase and SRC. And the question is then, is TRAF6 involved in the activation of these pathways as well? In order to elucidate this, Anahita Hamidi used prostate cancer cells and stimulated them with TGF-Beta for 30 minutes, which she could show then induced ubiquitination of the regulatory subunit of PI3 kinase, that is P85. In the TRAF6 knockout cells, there is no such ubiquitination, telling us that the ubiquitin ligase involved there is TRAF6. we could also show that a downstream kinase, which is activated by PI3 kinase, AKT or ACT, is also ubiquitinated in response to TGF-beta stimulation after 30 minutes. But of course, if we knock down P85, this doesn't happen. And this correlates also with the activation of AKT, which is seen as phosphorylation of serine 473, as well as threonine 308. So the TGF-beta induced ubiquitination of P85 alpha is not dependent on the kinase activity of the receptor, as is shown here, because if we introduce an inhibitor of the type 1 receptor kinase, it has no effect on the ubiquitination of P85, whereas it completely inhibits the phosphorylation of SMAD2. So we then wanted to identify the acceptor lysins that are ubiquitinated, and we could show by study of the various fragments of P85 that the region in between the two SH2 domains accepts ubiquitin moieties, and we then looked at lysins in this region, and two of these lysins had mutation of these two lysins had no effect, whereas mutations of two other lysins, 513 and 519, depleted ubiquitination of P85 dramatically. We then looked at an in vitro PI3 kinase assay. We measured the formation of PIP3, and as you can see, the wild-type receptor can mediate TGF-beta induced formation of PIP3. If one mutates one or the other of these two ubiquitin acceptor lysins, there is a suppression of this effect, and if you mutate both, there is a suppression of almost complete suppression. And this goes hand in hand then with activation of the downstream ACT, as you can see here, phosphorylation of serine 473 goes down to almost completely, as well as the phosphorylation of threonine 308. We then looked at invasion. And in order to do so, we first, in the control cells, looked at the effect of transfection of P85 wild type. As you can see, TGF-beta induces migration of cells, and this is enhanced by transfection of the wild type receptor. We then knocked down the wild type P85 molecule, and then we reintroduced either the wild type or these lysine mutant P85 molecules. And as you can see, the transfection of the wild type receptor causes migration induced by TGF-beta stimulation. And that is true also for the transfection of either of the two with one of the ubiquitin acceptor sites mutated, but not if we mutate both of them. You see that the migration ability of the cell goes down dramatically. So this links then the migration of cells to the ubiquitination of P85 and therefore activation of the PI3 kinase. Is this of any importance for patients? Well, using then sections from prostate cancers, tumors, we could see that first of all, with the more malignant tumors, Gleason score 8 or more, there is a lot of activation of TGF-beta signaling measured as phosphorylation of SMAD2 compared to sections from less malignant prostate cancer tumors, Gleason score 6. We could also see more phosphorylation of AKT in the more malignant tumor cells. Question is this linked to TGF-beta stimulation? Sorry. In order to address this, we took advantage of an assay called proximity ligation assay. In this case, we used two antibodies, one against the P85 in this case, and the other against the ubiquitin. And by adding these two together with the appropriate reagents, we can see if the two antibodies come close enough to each other, we see a signal, as seen by a red dot here. And as you can see, there is plenty of those in these malignant prostate cancer tumors and much less in the more benign, telling us that TGF-beta induced ubiquitination of P85 and thereby activation of PI3 kinase happens in the more malignant tumors. So this is the schematic illustration of the mechanism whereby TGF-beta induces PI3 kinase. the receptor complex, the tetramer of type 1 and type 2, is assembled by ligand binding. Together with the type 1 receptor, the ubiquitin ligase TRAF6 is located, and this then attracts P85 regulatory subunit of the PI3 kinase molecule, which is ubiquitinated in a region which is involved in the activation of the catalytic subunit P110, therefore inducing PI3 kinase activity. And it also attracts AKT, which is an important downstream effector in the PI3 kinase pathway, and AKT is also ubiquitinated and activated, as seen by phosphorylation at these two residues. Now, what about SRC? So SRC is activated by TGF-Beta in different cell types. This is 293 T cells. This is prostate cancer cells. And this is mouse embryo fibroblasts. And as you can see, after 30 minutes or so, SRC is activated, as shown here by an antibody recognizes phosphorylated tyrosine 416, which correlates to activation of SRC. In parallel, then, SMAD2 is phosphorylated, as is seen here, and SMAD3. So is this then dependent on the kinase activity of the receptor? The answer is it's not dependent on the kinase activity of the type 1 receptor, because if you use here a kinase dead receptor, it can mediate activation of SRC equally well as the wild type receptor and the kinase constitutive active kinase of the receptor. And then the question is whether it involves TRAF6. And the answer is no, because the wild type receptor kinase mediates activation of SRC, but that is also mediated by the mutant receptor, which is unable to bind TRAF6, as you can see here. So SRC is also known to be involved in invasion, but it is not dependent on TRAF6 after activation of TGF-Beta receptors. So this is the cartoon we have for the moment of the known non-SMAD signaling pathways involving then TRAF6 as a central player, but there is also pathways which are not activated by TRAF6. And many of them are involved in mediation of invasion and metastasis. This is pro-tumorigenic effects. Finally, I'd like to spend a few words about EMT. So in collaboration with Aris Mustakas, we have been interested in elucidating the effect of TGF-Beta induced EMT on metastasis of tumors. And as you can see here, as illustrated on this slide, EMT is seen during the development as a perfectly normal thing. It is also seen in wound healing, which has an important role, and then it is captured by cancer cells. And the EMT in the cancer context promotes migration of cells and formation of metastasis. So TGF-Beta induces EMT, as is seen here, on the conversion of this epithelial-like morphology of cells. This is staining for E-cadherin, which is an epithelial marker. After TGF-Beta stimulation, this sort of cobblestone staining is gone. There is still some staining, but it looks completely different. If we now look at the shape of cells by staining for actin filaments, you see that this cobblestone pattern is changed to cells become much more elongated, looking much more like fibroblasts. So we have been interested in learning what are the mechanisms involved in TGF-Beta induction of EMT, and through work from us and from many other laboratories, transcription factors of the snail, slug, and twist families have crucial roles in this process, in downregulating at E-cadherin and upregulating mesenchymal markers. But we want to see whether we can find something druggable that we could sort of use in order to prevent TGF-Beta-induced EMT. So therefore, we embarked in collaboration with Marino Serial in the screen, in which we screened the low molecular weight compounds, the chemical library of 17,000 components, for EMT induced by TGF-Beta. And this is the assay we used. Staining in red here is for E-cadherin, and as you can see, after 24 hours of TGF-beta stimulation, this goes down, and even more so upon prolonged stimulation, and after 72 hours, very little E-cadherin is there. Fibronectin, which is a mesenchymal marker, stained in green, in parallel then comes up. So we know that EMT is dependent on SMADs, thereby it is dependent on the kinase activity of the type 1 receptor. So we, as a proof of concept, we use a kinase inhibitor to try to revert EMT induced by TGF-beta. And as you can see, this is possible. The longer the inhibitor is there, the more epithelial the cells become. So using this assay with help of Andrew Shaw, who provided the chemical compounds, we obtained 16 hits, and we were able to lay our hands on 13 of those, and we then evaluated them in terms of their ability to inhibit TGF-beta induced EMT. And I'll show you data here only from 2 of them. We call them EPM, epithelial plasticity modifier, 10 and 13. And as you can see here on this slide, TGF-beta induces EMT of these breast cancer cells, and EPM13 reverts this, at least partially, as well as EPM10 likewise. This is the kinase inhibitor, which also then reverts the TGF-beta induced EMT, seen here as staining for cadherin, and also the shape of the cells by staining for actin filaments. You see the TGF-beta induced EMT is reverted by EPM13. It's also reverted by EPM10, but it has a completely different effect on the shape of cells, as you see here on this slide. So finally, if we want to develop TGF-beta antagonists for treatment of advanced tumors, it is desirable to inhibit TGF-beta's pro-tumorigenic effects involving EMT, invasiveness, and metastasis, while leaving TGF-beta's tumor suppressor effects unperturbed, involving then growth inhibition and apoptosis. So the possible approaches we have elucidated would be, on one hand, to inhibit the formation of the intracellular domain of the receptor, and we are currently trying to do this by antibodies. Alternatively, one can inhibit the TGF-beta activation of PI3 kinase, or inhibit TGF-beta induced EMT by the low molecular weight compounds that I just described. I should be honest and say that the compounds we have elucidated so far are not potent enough to be used in vivo analysis. We are trying to find better compounds. One could also inhibit the delta NP63, which I haven't had time to talk about, or inhibit the AP1 SMAD interactions. And we are trying to sort of see whether we can find ways of achieving this, and then the next step would be to test this in mice models. So finally, I'd like to thank the people involved in the work. This is the present group members in my group, and those who are marked in red have been involved in the work that I described today. And these are our collaborators. Again, those who are marked in red have been involved in the work I presented here, and former colleagues here, which have been involved also in the work that I presented. Thank you very much.
Thank you very much, Kali, for this wonderful journey through the complexity of TGF-beta signaling. I think we have time for a few questions, if there are some questions in the audience. I was intrigued, Kali, by this intracellular domain that you show is cleaved off only by prostate cancer cells. So I was wondering, so you show it happens in prostate cancer, not in normal prostate. What about other tumors? Is it a general phenomenon in all cancer types, or is it limited to prostate?
We have looked at a number of epithelial tumors and seen that the nuclear accumulation is fairly general. With regard to the cancer specificity versus normal cells, I don't know. And as I said, we don't really know the basis for this. It could be that the first cleavage just outside the cell membrane, which is done by metalloproteases, happens more easily in tumor cells that has more protease activity. But this is a speculation.
Sophie has a question.
How do these EPM, these inhibitors of EMT, in fact, act on which pathway? Do you have an idea of the molecules that they target?
This is, of course, a crucial question. And this is a black box screen. We don't really know the target. Our plan is to try to refine these compounds to get sort of useful compounds for in vivo studies. And when we have those that are good enough, we will use them as a bait, in order to, by a proteomic approach, identify the molecule it binds to. But we haven't come that far.
Other question? Don't be shy. Yes?
Hi, I had a question concerning the EMT that you described. Did you already investigate other factors from the TGF-Beta family, like BMPs, which are also known to be quite important in the EMT? And
yes.
Can perhaps be one of the answers to those inhibitors?
This is a very relevant question, and this is something which we are doing right now. There is often a sort of a dichotomy between TGF-Beta and BMP. Often they do opposite things. So BMP could actually revert EMT in certain contexts, in certain tumors. But we have seen several examples from several tumor types, like ovarian cancers and endometrial cancers, that also BMP are inducing EMT. So the role of BMP in members of the BMP family in EMT is complex, to say the least. And we are trying to sort this out.
Yeah, because in breast cancer, for example, BMP2 is quite highly expressed, and there it induces EMT with a role with CD44. So the two are linked. And yeah, perhaps this one.
So it induces EMT in breast cancers as well?
BMP2.
Mm-hmm.
Yes. Yeah. And then I had also a question concerning the proliferation of the TGF-Beta. So it acts as a tumor suppressor, but do you know which ligands can be important for the activation of the proliferation? Because in certain cancer, for example, SMAT4 is inhibited and induces then higher proliferative index. So do you know which ligands can be important to induce proliferation in epithelial organs?
Mm-hmm. Well, to start with TGF-Beta itself, it is, as I said, expressed at high levels in all solid tumors, as far as is known. And it is activated into tumor and suppresses growth initially. But during tumor progression, the tumor cells come to a point where there is a switch, and the cells loses the ability to be inhibited by TGF-Beta. And exactly why is not clear. This is also a very crucial question to try to answer in the field. With regard to other growth factors that can drive the growth of these tumors, I think this varies from tumor to tumor. There is plenty of candidates of the EGF family, the PGF family, and many other families of ligands.
Okay, thank you very much.
Other questions? Well, if there's no other question, I'd like to finish off and saying that you're all welcome to invited to a drink that will take place in the hall of the Duve Institute, ICP, 150 meters from here. There's some indication to find your way. And I would like to thank very heartfully Kali for this lecture and for being with us today. Thank you.